everyone, welcome back to another biology video. My name is Amanda and today I'm going to teach Biology Form 4 Chapter 3 Subtopic 3.3 Movement of Substances Across a Plasma Membrane in Living Organisms. So this part, let's talk about it from the textbook. Please open your textbook, page 57. Okay, so in this page, they will talk about the active and passive transport in living organisms. Basically, examples of the transports, active and passive. I already teach that in the last video. So example of passive transport. Passive transport is transport of substances through the plasma membrane without energy, no ATP. So, first example, gaseous exchange between an alveolus and a blood capillary through simple diffusion. So this one we learned in Form 3. You can see the diagram down here. It's the alveolus and the blood capillary. So the oxygen and carbon dioxide will go in and out through simple diffusion. So the oxygen from the alveolus will go into the blood capillary through simple diffusion. And then the carbon dioxide will go from the blood capillary to the alveolus also through simple diffusion. Okay, so the second point, reabsorption of water occurs when osmosis through the renal tube of the kidney. So in our kidney, there's a thing called renal tubule. So that one, there's a thing that happens where it absorbs water. So they will absorb water using osmosis. Okay. Third point, absorption of water by plant root hair cell by osmosis. So you see the picture down here, there's this plant. So the water and minerals will go into the plant's punya roots through osmosis, of course. And then last point, absorption of fructose molecule in the villus by vacillated diffusion. So the fructose molecule inside your intestine, there's this thing called the villus. So the fructose, it will get absorbed through vacillated diffusion. That means it's helped by proteins at the plasma membrane. Okay, next is active transport. Active transport is a transport across the plasma membrane, but they use energy. They use ATP. So first point, absorption of glucose and amino acid in villus. Second point, reabsorption of glucose through the renal tubule in the kidney. Okay, third point, transport of sucrose from a leaf to a phloem tissue. And the fourth point, absorption of mineral ions by a plant root hair cell. Okay, so for the plant root hair cell just now, water goes through osmosis, and then the mineral, they go through active transport. Okay, let's go to isotonic, hypotonic, and hypertonic. Okay, so you see here, there's three, isotonic, hypotonic, and hypertonic. So first, for isotonic, it's quite easy to remember. Oh my god. It's quite easy to remember. So iso has the letter S here. So S means same concentration. See, isotonic, same. So okay, isotonic means same concentrations of solution. Okay, so I drew beaker so it's more easier for me to show the example. So for isotonic is same concentration of solution. So this is the concentration of solution. Okay, you see? There's 2 on this side, 2 on this side, so they're both the same. For isotonic, solution A and solution B are isonic towards each other. They are isotonic towards each other because they have the same concentration of solution. So the water does not move. The water just stay because it's same concentration. They don't need to move, so they just stay. So next is hypotonic. Hypo rhymes with low. So hypotonic means low solute concentration. Hypo, remember hypo rhymes with low. So hypotonic is low solute concentration. In other words, it has high water potential. Okay, that means it has more water. So for example, look at A. A has low solute concentration. B has a higher solute concentration. So the movement of water is this way because A is hypotonic to B. A is hypotonic to B, so the water will go to B through osmosis. So isotonic means same concentration of solution. That means A and B has the same concentration, so they don't have to move. The water doesn't need to move. But for hypotonic means low solute concentration. A is hypotonic to B because A has a lower solute concentration to B. So the water will go to B through osmosis. And then next is hypertonic. Hypertonic. High sounds like high. So hypertonic is high solute concentration. Oops. Okay, so for this one, I'll draw the little moles here. Okay, now look at this diagram. A, this side has more solutes than at the side B. Okay, it has substance A or solution A has higher solute concentration than solution B. So the water will go which way? The water will come from B, go to A. For this one, from A, go to B. Why? Because this one, A, is hypotonic to B. So that means A has low solute concentration and B has high solute concentration. Like what we learned in the last video, osmosis means water going from a high concentration of water to low concentration of water, or a low solute concentration to a high solute concentration. So you see here, the water will go from low to high, low solute to high solute. This one goes from low solute to high solute. So So for this diagram, A is hypotonic to B, and for this diagram, A is hypertonic to B. But this one can be B is hypotonic to A, and this one can be B is hypertonic to A. This one, their solution A and B is isotonic to each other. Okay, now we are going to go to the effects of isotonic, hypotonic, and hypertonic to animal cells and plant cells. So first, we're going to animal cells. So inside the textbook, they used red blood cells. So up here, this little down here, cell cytoplasm contains solutes such as glucose and mineral salts. The fluid found in cells is known as intercellular fluid, while each cell is surrounded by extracellular fluid. So basically, this is our red blood cell. Okay? So, inside the cell has intracellular fluid, and outside here is the extracellular fluid. So, if the solution is isotonic to the red blood cells, then the water will diffuse into and out of the cell by osmosis at the same rate. So the water in the extracellular and the water intracellular, it will diffuse in and out the same, because it's isotonic, same concentration. Okay, so there is no net movement of water across the plasma membrane, and the cell maintain their normal shape. So the shape will be normal lah. It won't change shape. It will be like that, like healthy normal cell. But what if the cell is in hypotonic? Okay, so extracellular fluid is hypotonic to the intracellular fluid of the red blood cells. So hypotonic means that the hypotonic solution has low concentration of solutes, right? So that means there's more water inside the extracellular fluid. So what will happen is that the solution will be hypotonic to the red blood cells, right? So the water from outside will go into the cell. It will just keep going in because they want to reach the dynamic equilibrium. They want it to be even. This one already even because it's same. This one outside has more water than the inside, so the water will go into the cell. So what will happen is the cell will literally explode. Okay, the cell will swell and burst. Okay, so the red blood cell, it will swell. Okay, why does it swell and burst? So this is because the plasma membrane is too thin to withstand the osmotic pressure built up in the cell. So if a lot of water go in, the pressure will be higher, but then the plasma membrane isn't that strong enough and it's thin, so it will cause the cell to swell and burst. This thing, the burst of red blood cell, is known as hemolysis. Okay, so basically in hypotonic, the water will diffuse into the red blood cell. The red blood cell will swell and burst because the plasma membrane is thin and not strong enough to handle the pressure. So this whole scenario is called hemolysis. Hemolysis or hemolysis, I don't know how to pronounce. So, and lastly is hypertonic. So if the red blood cells are placed in a hypertonic solution, the extracellular fluid have less water than inside the cell. So the water inside the cell will go out. This one go in. This one will go out. Hypotonic, it go in, and hypertonic, it will go out. So what will happen if the thing go out? This is what happens. So the water go out already. The inside has no more water. Okay, not no more water. There will be less water. So the cell will shrink. In this one, the water go in, the cell will swell and burst. But for hypertonic, the water in the cell will go out, and then the red blood cell will shrink. So this scenario, or this thing that just happened, will be called crenation. Okay, so isotonic, they have same concentration, so the water will diffuse in and out in the same rate, so the red blood cells will be normal. For hypotonic, they have low concentration of solutes outside of the cell, so that means there is more water. So the water will diffuse into the cell through osmosis, and it will make the cell swell and burst. So this is called hemolysis. But for hypertonic, the solution outside has high concentration of solutes, so the water inside the cell will go out because outside has less water, and then this will cause the cell to shrink, and this is called crenation. Now we will go into the effects of plant cells when it goes into the solution that is isotonic, hypotonic, and hypertonic. So for isotonic, so basically the water potential will be the same. The concentration inside and outside the cell will be the same. Wait, inside and outside the cell will be the same. So like just now, the water diffusion in and out of the cell will obviously be the same. Okay? So it will be normal. So for hypotonic, the water potential outside of the cell, the extracellular fluid, the solution, it has more water because it's low. Low concentration of solutes. So there's more water outside. So the water will diffuse in, and that will make the vacuole like super big. Because we learned in chapter 2, vacuole holds water, right? So this is the plant cell. The vacuole expand and it pushes the cytoplasm and plasma against the wall. It will push the cytoplasm and stuff against the wall. So, but the plant cell don't burst because the plant cell has a cell wall that is strong and rigid. So, what will happen is that the cell will be turgid. So yeah, the cell will become turgid because water go in since it's hypotonic solution. There's more water out. So it will go into the vacuole. The vacuole will swell and then it will push the cytoplasm and plasma membrane towards the cell wall. But the cell wall is strong and it works to maintain shape, right? So I guess the shape is kind of maintained, but the only thing is that the plant cell is turgid. Okay? So for hypertonic, the outside solution has a higher concentration of solutes, which means there is less water outside. So the water will diffuse out of the cell, and this will cause the vacuoles and the cytoplasm to shrink. This will also cause the plasma membrane to be pulled away from the wall. So this process is called plasmolysis. So this will cause leaf and stems to bend downwards and wilt. This is how plants wilt. Okay, so for isotonic, inside and outside is same. Inside and outside is same, so the water will diffuse in and out. Same also lah. So it will be normal. For hypotonic, water will go inside the cell and then the vacuole will become swell. The vacuole will swell up and then it will push the cytoplasm and the plasma membrane towards the cell wall. So this causes the plant cell to be turgid. And for hypertonic, the water will diffuse out of the cell, causing the vacuole and the cytoplasm to shrink. This will pull the plasma membrane away from the cell wall, and this thing is called plasmolysis. This also happens. This is the thing that happened. This plasmolysis, this causes the leaves and stem to wilt downwards. You know. See, it's wilted downwards. That is all for 3.3. There's another subtopic, but its application in daily life. So I think it's better if you just read it yourself. So that is all for chapter 3. For the next video, I'm going to do for chapter 4, which is the biomolecules. What's the title again? Chemical composition in a cell. So I will teach that in the next video. Thanks for watching.